RNA-based parts encode functional RNA molecules or useful regions of a larger mRNA molecule that encodes a protein. Ribosome binding sites are RNA sequences that bind to the 16S rRNA and cause the initiation of translation. The ribosome binding site has two regions, one that binds the 16S and another that is the first AUG codon or start codon of the protein. Typically, these are spaced around 8 base pairs apart, but as you can see for this panel of arbitrarily chosen E. coli genes, the spacing is variable, and the precise sequence is also variable. To a first approximation, the more similar the orange region, called the Shine-Delgarno sequence, is to the 16S sequence, the higher the rate of translation initiation. However, gene expression levels are determined by multiple factors. Also note that not all start codons are AUG. The third gene in this list is a GUG start codon. GUG starts happen in around 8% of prokaryotic genes, and UUG start codons happen around 1% of the time. How best to describe an RBS sequence as a part remains a topic of discussion. Should it be the sequence up to the start codon? Should it include the start codon? How far upstream to the 5' end should it extend? To what degree can it be considered in isolation of its downstream sequence? Though there has been some study of this problem, it remains an important and unresolved question. Other RNAs encode more nuanced molecules. Ryba Regulators are RNA elements that bind other RNA elements and control gene expression. Often they work by changing the conformation of the RNA and thereby reveal or occlude a ribosome binding site. In other cases, they affect premature termination of transcription. tRNAs can also be encoded as parts. For example, UAG stop codons can be recoded as, say, a serine codon by introduction of an engineered tRNA into the cell. By changing the anticodon of this tRNA to CUA, the cell will begin translating UAG codons as if they were sense codons. Such tRNAs are called suppressor tRNAs because they suppress the phenotype caused by UAG mutations in genes. RNAs can also perform the non-covalent and covalent reactions observed more commonly of proteins. Aptamers are RNAs that bind to small molecules or proteins. They are functionally similar to antibodies. Ribose switches are like aptamers in the sense that they bind to ligands, but upon binding they undergo conformational changes that affect a downstream protein coding sequence. They do this via occlusion or exposure of a ribosome binding site or by causing premature termination. There are also coding RNAs that are catalysts that do chemical reactions, and these are called ribozymes. There are also short RNAs used to downregulate gene expression. In prokaryotes, there is a phenomenon in which binding of one RNA to another can occlude a ribosome binding site or stimulate cleavage by a native nuclease. In eukaryotes, this phenomenon is catalytically enhanced. siRNA, or its genetically encoded variant microRNA, are small 20 to 25 base pairs in length duplex RNAs that are used by higher eukaryotes to direct cleavage of homologous RNA sequences. One final class of important RNA parts are terminators. These sequences form hairpins which cause transcription to end. They come in two types, row-dependent and row-independent. They vary in strength, and like ribosome binding sites, this strength is complicated by other factors such as secondary structure. When combined together, an RBS, a protein coding sequence, and a terminator define the sequence of an mRNA molecule. When combined with a promoter part, a complete gene is generated.